Hi, my name is Noah. Behind me, you see a DNA structure. Okay, this is devoted to building 68 at MIT, which is the Koch building at MIT, which is devoted to bio research and the whole bio department here. This structure, the DNA, was not always obvious to everyone. Not everyone knew what we were made of and where we came from. But here today, you can see it's an emblem of where we come and how far we really have come. Thanks to two individuals called Watson and Crick and their use of the scientific method. I'll tell you about something called the scientific method. Okay, the scientific method can be decomposed into 6 different parts. You can see it right here. All right, and it's actually nonlinear. It's a cycle because the scientific method is something we're always working on. First step you're going to start with the scientific method is you're going to ask a question. Okay, this question can be about anything, pretty much. Let's say I'm curious to know what I'm made of. Okay, what determines my eye color? What determines how fast I walk? Whether I'm an athlete? Whether I'm academic? The question can come down to my DNA. And no one actually knew what this was. About 50 years ago, this concept was completely unknown. And 2 men called Watson and Crick, they wanted to figure out what DNA was. So what they did was they asked the question. That's where they started. The next step after you ask your question is going to be to make some observations. Alright, so what you always want to do before you jump in to ask a question, or rather explore your question, is you want to see what's out there. What's the research, what's the literature, what are people saying? Okay, for DNA specifically, there was some research out there. This guy, Linus, he actually thought that there was a structure for DNA called a triple helix. Okay, that was the structure. And then you do a little more research, you see that then there was this sort of critical breakthrough. Right? That was by Rosalind Franklin, and she had a photo called Photo 51, where she showed that there was sort of an X shape. There's actually no way that it was a triple helix. So before you go and continue with your hypothesis, which is what Watson and Crick did, you want to see what's out there, what are people saying? Sort of gather your research and figure out what the world is talking about. So building on your observations, you then want to construct a hypothesis. Okay, what you want to do in your hypothesis, and this is exactly what Watson and Crick did again, is you want to take what you've seen. Watson and Crick saw that Pauling had a triple helix. They said maybe a helix structure would work. Okay? Then they saw Photo 51, and they said, I'm not sure if a triple helix is right. It looks like there's an X shape. Maybe it's a double helix. And you can see here in the structure of DNA, okay, that you have adenine bonding with thymine. Okay, those are 2 nucleic acids. Cytosine and guanine. Okay, and here you can see there's an X shape forming. And that's characteristic of a double helix, not a triple helix. And that's what Watson and Crick noticed, and that's where they built their hypothesis. Alright, so I've got a hypothesis. Next step, to design an experiment. Okay? Critical step here in designing an experiment is getting a measurable outcome. Okay? I want to set up a scenario where I can put in a few inputs. Okay? Control that only those inputs are what's going to cause the output, and then measure that output. Okay, so the key here is to get something measurable. Okay, so for example, Watson and Crick did X-ray diffraction, okay, to see the image of the DNA. They were going to measure a shape. All right, so to this end, that's something measurable. You also want to control for certain things. You don't want there to be outside influences. So if I'm going to image something, I want to make sure that the lighting is not going to affect it. So if I do it in this room, or I do it in the next room, it's not going to matter. I need to control for external factors like that to make sure that the inputs I'm putting in the experiment will generate an output that I can rely on. That's what it's all about when you do an experiment.
Today, DNA experiments are conducted with highly advanced technology. These are polymerase chain reaction machines that amplify small pieces of DNA so scientists can analyze the genome of almost any organism. At the Broad Institute, machines such as hybridization ovens create the right conditions for DNA microarray sequencing.
So I've done my experiment. Okay, next step is going to be to collect data. This is the critical step. This is going to be what it's allowing me to see whether my output, which I measured, is giving me some useful information. So I've done my experiment, tally up the data, okay? I can generate graphs from this. I can pump it into equations and see if it's giving me the right output. All right, this is critical. All right, so you collect your data. Watson and Crick did this with X-ray diffraction. They saw that it would look nice, okay, in their very, very elaborate double helix model, which while elaborate was also very simple, okay? And then using that data, they verified that that was actually a valid model. Okay? And that was the critical step here. So you do the experiment to get the data to make a conclusion.
Here we see sophisticated graphs and high-tech machines, such as this genome analyzer, that help us analyze data that are collected in experiments designed by scientists today. Here we can see a genome sequencing and some more microarray analysis at the Broad Institute.
Now you're at your conclusion. This is really a happy place to be. You've done all the work you need to do. You asked your question. You did your background research. You made observations. Okay, constructed a hypothesis. Ran your experiment. Collected data. Made sure it verified. Now you can say, hey, look, I can explain to you what I'm made of. Why my eyes are brown or hazel, etc. Okay, so Watson and Crick did just that. In 1953, they published a paper called Molecular Structure of Nucleic Acids in Nature, a famous journal, and that's where they first unveiled the structure of the double helix, which, as you know today, is the predominant theory. Okay, it's the model we use for pretty much everything, and you can see that in the Human Genome Project, it's really taken off. Something recent called the Personal Genome Project, and all around MIT's campus, this is really sort of the hub of what's become of DNA, all because of the scientific method here, which Watson and Crick did step by step, meticulously, and they did it right, and they made a conclusion. Institute is one of the premier research institutes related to biology, genetics, etc. The person who founded it, Eric Lander, is actually the father of genomics. And behind me you see sort of examples of what the DNA discovery has led to. So that double helix that we talked about earlier, it's led to all these things, targeting cancer stem cells, blazing neutrinos to antibiotics. All these exciting things are being researched today, and we're answering these questions right here because of Watson and Crick's discovery using the scientific method.